Hello everybody and welcome to, let me adjust the camera there, hey there we go. Welcome to Monday, January 11th, 2021. Mr. Parrott here with our science lesson for today. Just a reminder, if you're confused about anything as I'm going through things, send me an email. If you'd want to participate in a Google Meet, those are always available too. You just got to email me and I'll send you a link and you can join up with the class if you'd rather try to access the class that way. That stuff is always available to you. Just a reminder that those things exist out there, but you got to initiate some of that stuff. Otherwise, I'm going to assume that these videos will answer a majority of your questions here. What we got going on today is that we're going to first do the bell ringer. That should be posted up there. And then we're going to do a little thing called RNA transcription. So we're going to do a little bit more of our notes today as we go day by day, going through things here. And we're going to basically build some RNA today at the end of it. So without further ado, let's look at our notes here. I'm going to skip ahead to number 4. This is what we did last week. We looked at different types of RNA. So we're going to look at the process of transcription. What is transcription, though? It is the process of turning DNA into RNA. And why is this so important? Is because RNA is what the ribosomes need in order to make our protein. Why are proteins so important? Is because we are made out of protein. All the different traits and characteristics are there because they are made using proteins. So we definitely need them to be created, and DNA has those instructions for what needs to be created as far as proteins go in every single one of our cells. So that's why it's important. The steps of how this works are very similar to the steps of replication that we did the other day in class. Now, this part was a little bit tough for us to understand as far as our activity goes, so I'm going to break it down a little bit more for us here today when it comes to the activity. But first, what needs to happen is that the DNA that's all coiled up and wound up, it needs to unwind and then unzip, which means to spread apart even further there. What then happens, instead of getting DNA pieces to combine up to one side of the DNA, we are going to get RNA pieces to come in and join up with one side of the DNA. Kind of similar to what is happening in the picture there. Notice that only one side is being connected to. Only one side is going to be connected to when it comes to making RNA, because remember, RNA is only single-stranded. Remember, though, when we're doing this, the weird change that is going to happen is that the thymines do not exist for RNA. The trees that the apples went in are cut down. There are no more trees, but instead now the apples are under those trees. They are underneath of them. So the T's are always replaced by U's in this case going forward, and you'll see kind of how that works when we build out our model here. We then get produced an mRNA, a messenger sequence, and messenger RNA is produced, and then that leaves, and the DNA just twists back up like it wasn't even untwisted to begin with. So the DNA is left untampered with, and we get a messenger to come out of it in the end. And that's it, because then we got number 5 going in right after that. So we're only looking at this small little bit here. So let's go to our activity and spend a little bit more time explaining how this activity will go. So this is our transcription activity. It is going to work similarly to the other day with our DNA activity when we did replication. Except I am going to modify your instructions for being on quarantine right now. So here's our color key of what everything is. We have our deoxyribose sugars, we have our ribose sugars, we have phosphates in there, and then we have all 5 of our nitrogen bases. Adenine, thymine, guanine, cytosine, and now uracil, which is our new one for RNA. What we're going to go ahead and start building out an RNA model here. And what you're going to do as far as the activity is that you're going to build the RNA off of what the DNA is. So what I am going to give you right now is your DNA template. Basically what this means is that this is how you are going to model your RNA after. This is going to be our DNA. It's going to be matching for it. So lay out up at the top the same exact configuration, because this is going to be the backbone of our DNA. It should go orange, purple, orange, purple, orange, purple, orange, purple, orange. There should be 5 of them in there. So like I was doing, drag them over and place them in. This is going to be one side of our DNA. This is the side of the DNA we're going to be making the RNA out of. We do this because this is the sugar phosphates that are going to be the backbone. Now, I think this was the part that was confusing people last week, if you were quarantining during replication, is that we need to have some kind of code to go off of. I am going to give you that code right now in this video. You need to place the following bases and connect them to the sugars like I am doing. The first one should be an A. The second one, we're going to have be a C. The third one, we're going to make a T, but notice that the T doesn't nicely fit together. So we need to rotate the T upside down so that it kind of fits into that little groove again. The next one we're going to have be a G. Once again, just like with the other one, notice that the G doesn't quite fit, so we need to flip it upside down so that the square matches the square peg. Then the last one, let's go ahead and add another A to the very end. This is going to be your DNA code that you are going to use to make your RNA out of. Now, here's what you're going to do. I'm going to show you the first step of this, and then you're going to finish it as a part of your assignment. We need to make the RNA. So notice up here we have this brown piece. This is our ribose sugar. We're going to add this to the bottom. The ribose needs to be flipped around so it faces the correct direction. So notice how it's got the little like pointy part of the pentagon. You got to flip that around to the other side so it's got the little other peg to it. You need to make an RNA strand out of the remaining pieces of the phosphates and the sugars. So because we're building an RNA model and it's the bottom strand, you're going to have to flip all these pieces around. Then what you are going to do is you need to find what side will match up with the A and place it on the RNA molecule. In this case, remember, I'm going to give you the first one here for free. Remember, when you're matching them up, you can go up here and see if they fit. The A and the U fit together, so down here, the first base would be a U, because the A and the U matches together when we're building RNA. So you're going to be matching up the bases, creating them the rest of the way. There is then some questions to answer. When it talks about the two sides, we're talking about the top side and the bottom side, the top half and the bottom half. So there's a couple of questions to answer up, 7 of them that go along with this one. If you have any questions at all about how this works, please email me. We can work something out. I can try to explain it to you. Otherwise, we'll wait and see what we come up with. I'll look at what you've submitted to me, and then we can talk when you come back of what we need to do to address the things that you're obviously missing because you're not in class and you're not able to answer those or ask those questions when they come up. But please do not hesitate to ask me those questions. Submit it to me when you're done. If you have any questions, email me. Otherwise, I hope to see you back in class real soon because it's just so much easier to have class when you're able to be a part of it, and I love that you're able to be a part of it. So I know a lot of you that were starting the year off on quarantine are going to be coming back this next upcoming week. So I'm so excited to see you back in class. So hopefully I will see you back sooner rather than later. But if not, hopefully you're taking care of yourself. And hopefully I will see you soon. See ya.